Okay, we're going to move on in this session to look at this third dot point here, which is using monoclonal antibodies to treat cancer. Okay, so this really relies on obviously your knowledge of how the immune system works, and also of how cancerous cells arise in the first place. So it might be a good idea to pause here for a second and just recap or think about these three questions. So, what is it that causes cancer tumors? What goes wrong, and why do those tumors develop? How do plasma B cells fight pathogens? And what is passive immunity? So pause and recap those things, and then we'll kind of go through those because they're all really important for understanding this concept. Okay, so hopefully you remember that cancer tumors occur when a cell is damaged, which happens quite frequently. DNA might be mutated or it doesn't replicate properly. But instead of being destroyed, like would happen over here, and normally a damaged cell would go through apoptosis, that message doesn't get received for some reason. Okay, and so what happens then is that those damaged cells continue to divide, as cells do, and eventually we get this kind of big mass of damaged, unhealthy cells, and that is what we call a tumor. Now, going back to what plasma B cells do, so when a pathogen is in the body, so a bacteria or a virus, B cells produce antibodies, these little guys down here. Those antibodies travel to pathogens in the body and bind to them, like this. So those antibodies are released from the B cells, and then those antibodies kind of flag those pathogens or make them more visible to other cells in the immune system to destroy them. And the last recap question, what is passive immunity? Remember that immunity itself is having specific antibodies for specific pathogens in your body. Passive immunity is when those antibodies are produced by someone or something else. So the example we've looked at is from a mother to their baby or through the injection of antivenoms. And these antibodies give immediate protection. They can straight away go and find the pathogens that they're looking for, but it is only temporary protection. Okay, monoclonal antibodies. Sounds like quite a big name, but when we go through this, it should make sense. These are antibodies that are produced in a lab. Okay, so they're an artificial thing. And the way they are produced, which we'll look at a bit more in a minute, but a B cell that is modified, which we'll talk about later, a B cell is made and then it is cloned. So we start with one cell, which is the mono part of that, and then that cell is clonal. So we get a bunch of clones of a cell that started from one cell. Now, because they are B cells, they produce antibodies. And because all of the cells came from one original cell, all of those antibodies will be identical as well. And this treatment, it can be used for a few different types of diseases, but what we're going to look at is in the context of cancer. So these cells or these antibodies can be used to treat cancer by binding with the self-antigens of those cancerous cells. And that obviously flags them then to the immune system. So over here we've got a cancer cell, so it's not a pathogen, but it is a damaged body cell that can cause a lot of problems. And these antibodies can go and bind to that cell and basically trigger an immune response to get rid of that cell. All right, the steps for making these you do need to know. So I've included this image for you to download. I really recommend that you kind of have this accessible while we go through the steps in a minute. I couldn't put them all on one slide, but the image along with the steps will hopefully make it a bit clearer. I'll print some of these out next week as well for you to add into your notes. So while you're writing down these steps now, maybe save, a third or a half of a page for the diagram to go with it. Okay, so making monoclonal antibodies. The first thing that needs to happen is we need to identify the antigen, the self-antigen, on the particular type of cancerous cell that we're trying to treat. So let's say, for example, looking at a melanoma, like a skin cancer, scientists would need to extract some of those cancerous cells and identify that antigen and isolate it, which basically means extract it, take a copy of it. Those antigens are now injected into a mouse. Okay, so almost like a vaccination. If you think about like a subunit vaccination, a mouse is vaccinated with the antigen of this cancer cell. Now, hopefully you're thinking ahead, what will a mouse or any mammal or organism do if injected with an antigen? It will go through an immune response. Okay, so the mouse goes through the immune response because of this foreign antigen and produces B cells, B lymphocytes. Those B lymphocytes are then extracted. Okay, so they're taken out of the mouse, and that's in the spleen. The spleen is the organ that kind of will hold a lot of those lymphocytes. So we take out the B lymphocytes, and those B cells are now fused with another type of cell, which is a type of cancerous human cell called a myeloma. Okay, so remember at the start I said that we're cloning a modified B cell. This is the modification. We've got these B cells that have been made by the mouse, and then they're extracted, and in a lab that is fused with a cancerous cell. And that fused cell becomes known as a hybridoma. Okay, so the B cell plus the myeloma equals a hybridoma. Now, because this cell is a fusion of a B cell and a cancer cell, it's got two really important properties. The first thing is that it can produce antibodies because it is a B cell. Okay, so that original B cell that was made can make antibodies. Also, because it is a cancerous cell, it is able to replicate really quickly. Okay, cancerous cells behave a bit differently. They divide really quickly. So what we're getting now is a B cell that can make antibodies that can also divide really, really quickly. And this is where we get our clones. Okay, so that cell is left to replicate or to clone, and then the antibodies that are made from all of those cloned cells are extracted, they're put into a serum, and then they are injected into the patient that has this particular type of cancer. Okay, so you're not injecting the B cells or those myeloma cells, you are injecting the antibodies, which makes it a type of passive artificial immunity. Okay, so we'll just revisit this picture again. So we've kind of isolated the antigen of the cancerous cell. That's injected into our mouse here. It's not going to cause any problems for the mice, just in case you're worried about that. So that's injected into the mouse. The mouse produces B cells. Those B cells are taken out of the mouse, mixed with these myeloma cells, and that produces hybridoma. And you can see there in this picture, the cell is kind of half yellow, half red. It's a combination of those two cells. And these hybridoma would be producing all of these antibodies. Okay, we have thousands of antibodies on each one of the cells. We let it clone. We've got heaps and heaps of identical antibodies, and they are known as monoclonal antibodies. They're extracted and then given to the patient. So what do those antibodies do then once they reach a cancerous cell? Okay, it is important to understand what happens then, how it actually treats cancer. So one thing, these are going to come up in a weird order for some reason. One thing that these antibodies might do once they've bound to a cancerous cell is trigger apoptosis. So tell that cell to destroy itself as it should be doing, but for some reason isn't. Another thing it may do is prevent the growth of the cell by blocking molecules that attract new blood vessels. So basically, it's stopping blood flow coming to the cells. The cells can't get the nutrients they need to continue dividing. So that will stop it growing. They can also be combined with particles or chemotherapy drugs to deliver molecules directly to the cancerous cells. So traditional chemotherapy involves injecting pretty heavy and nasty drugs into the body. And the problem with that is obviously they destroy lots of cells, not just cancer cells. Whereas if we can put those drugs basically on these antibodies that will only bind with the cancerous cells, it targets those cells and doesn't damage other cells as much. And I don't know why that came up last. But another very common thing that they do is just flagging those cancer cells as foreign so that immune cells like our phagocytes come along and help destroy them. It can also trigger cytotoxic T cells to destroy them the way that they should be doing. Okay, we'll hopefully get a chance to go over this again next week when we do a bit of revision before the SAC. But please go into the textbook, read the section on this. It's only 2 pages. Have a read of that and have a go at those questions about monoclonal antibodies. Bye.